Welcome back to the deep dive, everyone. And yeah, welcome to Green Dolphin Science. This is where we take all that source material, send our way the papers, the articles, your notes, and we really try to boil it down, find the most interesting bits, the important takeaways. Think of it as your shortcut to getting up to speed without wading through everything yourself. So today we're actually heading to the beach, metaphorically anyway. We're zooming in on Atlantic ghost crabs and how they connect to, well, a huge issue. Microplastic pollution. You know the scene, sandy shore, maybe some waves. But then picture these super fast little crabs almost blending in, right? And they're dealing with this invisible sprinkle of plastic everywhere.
Exactly. And the main source we're digging into is this really fascinating study. It was published not long ago in the Marine Pollution Bulletin, and it focuses squarely on the Atlantic ghost crab. Its scientific name is Ocypode quadrata. The big question they asked was, could this crab, which you see on so many beaches, actually work as a reliable biomonitor, specifically for microplastics in the sand? So yeah, our mission today is really to unpack all of that. What did the researchers actually do? What did they find out? Maybe some surprises in there. And, what does it mean? Can we use these crabs as little environmental detectives, or maybe not? And you might be thinking, why crabs and tiny plastic? Well, because it tells us something really important about our coasts, how pollution moves around, how it gets into the ecosystem. It's a bigger story.
Okay, let's paint a picture of these crabs first. Atlantic ghost crabs, Ocypode quadrata. If you've been on sandy beaches, particularly warmer ones, you've probably seen them, that flash of movement, right? They're super quick, often pale, almost see-through sometimes, which is where that ghost name comes from, I guess.
Yeah.
Darting into their burrows.
Right. And scientists have had their eye on them for a while, actually.
Oh, yeah? Why is that?
Well, they're considered pretty decent indicators of how healthy a sandy beach is. They're sensitive to things humans do, like driving vehicles on the beach or, building too close to the shore. Plus, they live over a huge area. They're not too hard to find and collect for studies. And the paper calls them one of the most conspicuous invertebrates out there. So yeah, they're already kind of known signals for beach conditions.
Gotcha. So they show the general health. But this study is about something specific. Microplastics.
That's right. Microplastic pollution, or MP. We hear a lot about plastic in the ocean, marine debris, and the source material highlights just how much is plastic, like 80 to 85% globally. It's staggering. And it doesn't just float around as big pieces forever. You've got waves, sunlight, abrasion. It all breaks that plastic down into smaller and smaller bits. These are the secondary microplastics, technically, anything less than 5 millimeters.
Like tiny fragments.
Exactly. And you also have primary ones, like industrial pellets, maybe microbeads, though those are less common now. But on beaches, a lot comes from larger stuff breaking apart. And the trouble is, these tiny pieces can sort of soak up other harmful chemicals. And crucially, for animals living right there, they're easily eaten. What this study points out is, while we know a fair bit about marine animals in the water eating plastic, we know less about the invertebrates living on the beach itself, even though beaches are, as the paper says, major hotspots for all that plastic washing ashore.
Right. It makes sense. All that ocean plastic has to end up somewhere. So this study is filling a gap then, looking specifically at the ghost crab. You mentioned they're good indicators, but this study tested them as biomonitors. What's the real difference there?
Yeah, that's a really important distinction they make. An indicator, it basically tells you about the general state. Are things okay, or is the environment stressed? Like, are the crabs even there? Are they behaving normally? A biomonitor, on the other hand, is more quantitative. The hope is that the amount of a specific pollutant inside the organism can tell you how much of that pollutant is in the environment.
Ah, okay. So can the plastic in the crab's gut actually measure the plastic in the sand? Is that the idea?
Precisely. That was the core objective. Is the Atlantic ghost crab a useful biomonitor for microplastic density and beach sediment?
How do they test that?
They set up two main hypotheses. Think of them as specific questions needing answers from the data. Hypothesis 1, or H1, was about the source. They asked, is the sediment, especially from the strandline, that line of seaweed and debris left by high tide where crabs often feed, is that the main place they're picking up microplastics?
Okay, how'd they check that?
They collected crabs and sediment samples from that exact zone. Then they meticulously compared what types of microplastics and in what proportions were in the crab guts versus in the sand.
Makes sense. And the second hypothesis, H2.
H2 tackled the biomonitoring question head-on. It asked, is the amount of microplastic inside a crab's gut related to the amount of microplastic pollution in the sediment on different beaches?
So comparing beaches with maybe different levels of pollution.
Exactly. They sampled beaches with different levels of nearby urbanization, thinking that might mean different plastic loads in the sand. Then they looked for a statistical link, a correlation. Do beaches with more plastic in the sediment also have crabs with more plastic in their guts? Consistently across places and time.
Okay, let's get into the results. What did they find for H1? Is the sand the source?
Well, for H1, the answer was pretty clearly yes.
Okay.
They found really strong similarities between the types and even the colors of microplastics in the crab guts and in that strandline sediment.
So that supports the idea they're eating it right from the sand.
Definitely. And the details are quite telling. The similarity was mostly down to microfibers.
Fibers, like from clothes.
Yeah, likely. Black microfibers were about 49% of the shared stuff. Blue ones were around 45%. And overall, fibers were the dominant type. 93% of the plastic in the guts, 90% in the sediment. Super high.
Wow.
So H1 was confirmed. The sediment they sift through while feeding on detritus on the surface, it is a major source. They're accidentally ingesting these common MPs.
Were there other types of plastic, too?
There were, yeah, but much less. Bits of hard plastic, about 2.5%, some soft plastic film, maybe 1.9%, and even tiny bits of polystyrene foam, like styrofoam, around 1.7%.
Yeah.
They also mentioned finding some bigger things occasionally, like bits of fishing line or bait elastic, which shows the crabs interact with larger debris too, even if it's mainly the micro stuff they're swallowing.
And those dominant fibers, black and blue, does the study say where they might come from?
It suggests likely sources are, synthetic fibers from washing clothes, getting out through wastewater, but also the breakdown of fishing gear. Think ropes, nets, that kind of thing. The colors black and blue fit well with common fishing materials and also textiles found in wastewater.
Okay, so point one confirmed. They eat plastic from the sand. Now for the big one, H2. Does the amount in the crab match the amount in the sand? Like, more pollution on the beach equals more plastic in the crab.
Right, that's the crucial biomonitoring question. And here's where it gets really interesting.
Uh-oh. Sounds like a twist.
Well, the study found no relationship. H2 was not accepted.
No relationship at all. That seems weird.
It was surprising. Looking at their data, the percentage of crabs that actually had any plastic in them varied massively. On some beaches, maybe at certain times, it was 0%. On others, literally 100% of the crabs had plastic.
Okay, huge variation.
But, and this is the kicker, the actual density of microplastics they measured in the sediment, it was pretty much the same everywhere they looked.
Why, really? Even on beaches near cities versus more remote ones?
Yeah, it stayed relatively constant, hovering around 0.1 to 0.2 plastic items per gram of sediment. So you didn't see this pattern where high sediment plastic beaches consistently had high gut plastic crabs. The two didn't track together.
Huh. So finding plastic in a crab doesn't tell you how much plastic is in the sand right there.
Not reliably, according to this study, at least not on the spatial and temporal scales they examined. Even though they get plastic from the sediment, the quantity inside them isn't a simple reflection of the quantity in that sediment.
Which means they're not great quantitative biomonitors for sediment pollution, then.
Exactly. That's the major conclusion regarding H2.
So why not? If they eat it from the sand, why doesn't it correlate? What did the study suggest?
They explored a few reasons, mostly boiling down to the crab's lifestyle. One big factor is what they call high trophic plasticity.
Trophic plasticity. Sounds complicated.
It just means they eat a lot of different things. They aren't just sifting sand. They're scavengers, finding dead stuff. They're predators, catching other small animals.
Like they might eat a dead fish washed up.
Precisely. Or they might eat other invertebrates that have already eaten plastic themselves. So the plastic in their gut could come from the sediment, yes, but also from contaminated prey or carrion. Multiple potential sources, not just the sand patch the scientists measured.
Okay, that makes sense. Diet's too varied.
Right. And related to that is their behavior. These crabs move around a lot. They forage widely.
They don't just stay in one spot.
No, they cover different beach zones near the water, higher up the beach, maybe near lagoons behind the dunes. The sediment sampling focused on that strand line, but the crab's actual plastic exposure zone is probably much larger and more complex.
So sampling one spot of sand doesn't capture their whole world.
Exactly. And another thing is their opportunistic feeding. The study mentions they seem to prefer punctual and high calorific value food, meaning finding that dead fish is probably more exciting and rewarding than just sifting sand all day. So maybe their plastic intake is skewed by finding one really contaminated high-value meal rather than just steadily accumulating it from the background levels in the sand.
So a big plastic dose from one meal could throw off the average.
Potentially, yeah. It complicates the picture. Interestingly, the study did find one connection. Crabs with fuller guts were more likely to have plastic.
Just eating more in general means more chance of eating plastic.
Seems like it, which makes sense, right? But other things they tested, like the crab size or how much general food, organic matter prey, was available in the sand, those didn't predict plastic ingestion in their final analysis. Gut fullness was the key factor they found, besides the source similarity from H1.
Okay, so let me try and sum this up. They definitely eat microplastics, and the strandline sand is a key source. But because they eat lots of different things, move all over the beach, and jumped on big meals when they find them, the amount of plastic you find in one crab at one time isn't a simple reflection of how much plastic is in that one patch of sand.
That's a great summary. Their lifestyle is just too complex for them to be a straightforward, quantitative measuring stick for sediment pollution in this way.
How does this compare to other animals that are used as biomonitors?
Well, the classic examples, especially for water pollution, are things like filter feeding mussels or clams. The study conclusion even mentions potential beach filter feeders, like Donax clams or Emerita mole crabs, might be worth looking at for comparison.
Why are filter feeders often better for this?
First, they process huge amounts of their surrounding medium water, in the case of mussels, so they're constantly sampling what's suspended in it, including microplastics. Second, like the ghost crabs and sediment for H1, filter feeders often do ingest plastic types in proportions similar to what's available around them.
Okay, but what's the key difference then?
The key difference is often that H2 result. With many filter feeders, studies do find a clear, more direct relationship between the concentration of a pollutant in their bodies and the concentration in the environment. Because their feeding is more consistent, less varied, their internal load tends to track the external load more reliably.
Got it. Consistent filter feeders versus opportunistic, wide-ranging crabs.
Basically, yes. Different tools for potentially different jobs.
So wrapping up the ghost crab's role here, what's the verdict from this study?
The verdict is they are valuable indicators. Finding plastic in them confirms plastic presence and proves it's entering the food web on those beaches. So they are good qualitative indicators telling you that plastic is a problem.
But not quantitative monitors.
Correct. Based on this research, they are not reliable quantitative biomonitors for tracking the specific level of microplastic pollution in the sediment across different sites or times, because their complex feeding and behavior muddy that relationship. And the researchers themselves suggest this highlights the complexity. Effective biomonitoring might need a whole toolkit, maybe using multiple species with different habits, like comparing these ghost crabs with filter feeders on the same beaches in future work. That could give a much richer picture.
Which brings it back to you, our listeners. I think this is a great example of how science works, right?
Right.
Trying to find the right tools and understanding the details of animal behavior is crucial. It shows how pollution interacts with wildlife in really intricate ways, all shaped by how an animal actually lives its life. And figuring that out helps us get better at measuring the problem and hopefully eventually tackling it.
Absolutely. It's about choosing the right lens to view the problem accurately.
So here's something to think about next time you're maybe walking on a beach, maybe you even see one of those elusive ghost crabs. Consider its busy life, scavenging here, hunting there, digging, running. How does that complex behavior influence what pollutants it encounters compared to, say, a clam buried in one spot filtering water? It's not always a simple path for pollution through an ecosystem, is it?
Definitely not. And it reminds us that scientists need to be quite clever and very specific in choosing which species and what aspects of their biology will give us the most accurate environmental insights. It's a constant process of refining our methods.
A great point. Well, thank you for joining us for this deep dive into the world of ghost crabs and microplastics. Keep an eye out for them on the sand, and remember there's a lot going on beneath the surface. From the deep dive, we'll catch you on the next one.